This whole process of meiosis and sexual reproduction is really important because it produces a huge amount of genetic variation. And genetic variation is really important in a population because it allows that population to undergo evolution. And we'll talk about that, how that's all related in our next unit. But I wanted to make a video that is specifically on those sources of genetic variation and where it all comes from in this process of sexual reproduction. Because this genetic variation is one of the features that we don't see in asexual reproduction. In asexual reproduction, individuals are basically a clone of their parents. There's no variation. And there are some advantages to that. We'll talk about that a little bit later. But sexual reproduction and the process of meiosis are really important because of the genetic variation that occurs. This random picture is mostly because I needed a picture there, and it's just supposed to represent sexual reproduction. I'll let you use your imagination there. But anyway, the first source of genetic variation is mutation. So we talked about mutations a little bit in our last unit. Mutations arise from a variety of sources. They can be environmental, so x-rays, cigarette smoke, things like that. Errors occur in DNA replication. So replication is really good, but it's not perfect, and sometimes errors occur there. Now, if we're talking about heredity and sexual reproduction, the only mutations that we're really interested in are those that occur in the gamete, because those are the only ones that are passed on. So if you acquire a mutation in a cell, in a somatic cell, it's in your lifetime, it's not going to be passed on to the next generation of offspring. But if something happens and your gametes acquire a mutation, and then that gamete fuses with a sperm or an egg, depending on the situation, that genetic mutation can be passed on. Now, we talked about earlier about some mutations being good, some mutations being bad. But either way, a mutation is a new source of genetic information that has potential implications for the next generation. So our next source of genetic variation is crossing over. So I talked about crossing over a little bit in the last video during meiosis. Now, the reason for crossing over is to hold homologous chromosomes together as they move to the metaphase plate. Now, this ensures that they are separated properly so that each cell at the end of meiosis has the right number of chromosomes and the right type of chromosomes. So crossing over is a quite complicated molecular mechanism that we're not going to go into. It's kind of interesting how it all actually works, but I just want you guys to know the general mechanism here. So we start out with our homologous chromosomes, our maternal and paternal chromosomes in their sister chromatid form. This is actually called the situation we're going to make is actually called a tetrad because we have 2 pieces of DNA from mom and 2 pieces of DNA from dad. And when they come together, they line up and basically hold hands in the cell so that when the spindle fibers attach to them, they can move together. Now, this isn't hand holding like you would hold the hand of a younger sibling or your mom or a girlfriend or a boyfriend where you'd let go and you keep your hands. This is like actually a physical exchange. It's like as if you switched hands and now because you held hands, now you have the other person's hand. I know that's way weird, but I'm just trying to explain to you what is actually happening here. So the sister chromatids pair up and they actually cross over. Now this cross that occurs is called chiasmata. Now chiasmata actually occur at several locations along the chromosome, and the more they are, the more stable, the more chiasmata there are, the more likely that chromosome is going to stay together. So eukaryotic chromosomes usually form 2 or 3 on each arm of the chromatid, but we're just doing 1 for simplicity. So they line up, they link up, they go to the metaphase plate together, and then at the metaphase plate they are pulled apart, and the result is that they leave behind some of their own information. So then when we get to the end of meiosis, and let's assume that each of these is a sperm or an egg, depending on the parent, you can see that each one is genetically identical. Now keep in mind that those outside ones, the ones that were on the outside, didn't exchange any information at all. So they are essentially the same as the parent. But these ones are not. They are new combinations of the parent's chromosomes. So crossing over doesn't actually create any new genetic information, not like mutations. Mutations created new information. Crossing over just makes new combinations of information, which can lead to novel traits in the next generation. So crossing over is really important in an evolutionary sense, just like mutations. Because mutations give us new information, crossing over gives us new combinations of information. And new combos of information can also be important in evolution. So whether it's a new allele or a new combination of alleles, they might give an advantage or a disadvantage to that generation of individuals. And so this is where evolution comes in, and where evolution begins to work on a population. Another source of genetic variation is random assortment. So I already talked about this one. You saw this slide in the meiosis 1 slide. So this is crossing over during meiosis 1. This is our random assortment, where we get any combination at the metaphase plate of maternal and paternal chromatids. And so when they're separated in anaphase and eventually telophase, you get, each cell has new combinations of maternal and paternal chromosomes on top of the crossing over. So again, we can see that there's variation just in this process, even before we get to the last step, which is sexual reproduction. So our last source of variation in this whole process comes from the actual fusion of gametes, where we get very different genetically eggs and sperm. So presumably the individuals that are causing this union between the sperm and the egg are not related, that their genetic information is much different because they come from different families. And so now you have any mutations that might have been introduced, any crossing over, any random assortment, plus new combinations from those parents that are also genetically different. And so you can see how quickly all of this adds up, and you can see the result of that if you look at the variation in a population just in our classroom, or just in Belleville, or just in the United States. And then worldwide, there's also a huge amount of variation, and all of that variation comes from sexual reproduction. So the process of meiosis in sexual reproduction produces male gametes and female gametes. The male gametes are the sperm, and they are basically produced via meiosis exactly the way that I just described. So sperm are made that way. Eggs are made that way with a slight variation. So for every every cell that's produced in an ovary, you would expect to get 4 eggs. But in reality, you actually only get 1. And the reason for this is that the egg needs a lot of nutrients, and the nutrients come from the cytoplasm. And so while the sperm is really small and motile, the egg is not. The egg contains a whole bunch of cytoplasm that's going to act as nutrients for the fertilized egg and then the growing organism. So the process of meiosis here actually occurs in the same way. We start with our diploid cell, we go through meiosis 1, we separate our homologous chromosomes, and then we lose one of the eggs. So if you'll look, you'll see that one of these cells is much, much bigger. This is because the cytoplasm is unequally distributed. Now, because this cell lacks cytoplasm, it's eventually going to sort of shrivel up and go away. That is called a polar body. Now, the other one, the remaining cell, is going to go through meiosis too. So it's going to go through, and now we're going to separate our sister chromatids, produce our other cell as part of that cell division. And since it doesn't have enough cytoplasm, it's actually going to shrivel up and go away. So in essence, you end up with one really big female gamete, a really big egg that has lots of cytoplasm, and the other three cells that you would, in theory, get from this division never happen. So this one should divide into two cells. Neither one of those survive. And then this one that came out of this second division goes away as well. So those polar bodies are degraded in the uterus, and the only one of them, this one, that the large one that has the larger amount of cytoplasm, is the one that actually could potentially be fertilized by a sperm. So if we look at this picture up here, the egg is much larger than the sperm there. And that's because the genetic contents of that sperm are going to fuse into the into the egg, and then it's the egg that's going to begin division for the growing organism.